Okay, before I get started here, a little disclaimer, because some of this is going to be talking about medical information. I'm not a doctor, and I don't play one, oh, here we go, on TV, but last weekend I did replace the motor in my Samsung dryer, so I am qualified to do that. Okay, with that out of the way, this is actually kind of an auspicious day because every year, April 25th, is National DNA Day. And why is that the case? Because about 19 years ago, actually at UC Santa Cruz, the final assembly of the human genome was done and uploaded to the internet by my boss, David Hausler, there on the right, who's the founder of the Institute, one of the top scientists in the world right now, and a grad student at the time, Jim Kent. And so they assembled it, and we'll explain what that means in a minute, and then they uploaded it to the internet for everybody to have, because we felt strongly that this piece of information is humanity's piece of information. So that was the first time we were able to really read our own source code, our own design, which is kind of an amazing thing to do. So what does that mean? We're going to do just a tiny bit of biology here, and some of you may have seen this from my last talk, but it kind of gives you some grounding here. Each of us has 46 chromosomes, right? We get half from mom, half from dad, sort of. I'm going to go into some details on that later on. 3 billion base pairs, so there's 3 billion letters in a row, 2-bit values, if you will. 25,000 genes in there. That's only about 1.5% of the genome. And a lot of people ask me, what's a gene? What does that all mean? So a gene generates proteins. The way to think about that, though, is even though only 1.5% of it is proteins, what's the rest of it doing? Corn, some species of corn have 100,000 genes, which is kind of depressing when we found out that we have less. So if I'm going to build a house, let's say I'm going to build a house and over there is Frank Lloyd Wright, right? And we both have a pile of bricks. The bricks are the genes, the proteins. Now I'm going to build the house. It's going to be kind of maybe safe. Frank Lloyd Wright's going to build it and it's going to be epic and beautiful. So it's not the genes per se, it's what we do with the genes. And the rest of the genome is what's doing that, regulating and kind of figuring out how the genes should be turned on and should be turned off. Any two of us differ by less than 0.5%. This is the clear and unambiguous definition of why we're all in this together. We may differ by politics, we may differ by religion, we may differ by taste in food and sports, but we're all in this together. This is the thing that binds everybody on Earth. And in fact, it not only binds all of us together, but if you look at that window, when you walk out today, look around, anything you see that's alive, a bug, a tree, a bacteria, everything we've found so far on this Earth that's alive is based on this DNA, this source code. Now we may find something, out in the globe, out in the universe somewhere that's different, but this is quite remarkable that this is the way the Mother Nature concluded it should all work. Every time there is a new baby, all the DNA came from mom and came from dad, but about 75 of those mutations are totally new, just totally random. And that's how Mother Nature kind of primes the pump for new evolution. Every day, you're generating about 250 billion new cells. Anybody here in storage? Right? You think about error correction, right? You have to somehow maintain information for billions of years. You need to copy it and fix it. This is remarkable. I think this is, if you kind of compare this to storage technology, not that storage technology is not amazing, but this is an amazing feat. And every time those get copied, there's about 0.7 mutations, 0.7 errors. Now, sometimes that's okay, but sometimes that's called cancer. Okay. So, what this talk is about is talking about kind of consumer genomics, kind of what's going on, where we're moving to now, and then where it goes in the future. And hopefully some of it will be somewhat personalized, so feel free, if there's something up there that's a burning question, stop me and ask a question, and I'll be around afterwards if you want to ask anything confidentially. So consumer genomics, Genomics 1.0 was really started when 23andMe came out and said, hey, for some couple $100, you can buy this kit, spit in a tube, and you'll get back some information. How many people here have either done this or had a relative that's done some genetic testing? Okay, because if anybody in your family's done some testing, then you've effectively done some testing. 1 in 25 Americans, actually it's probably higher now, has done testing. DNA testing kits were the #1 best-selling product on Amazon, other than Amazon branded products, on Black Friday, Cyber Monday, 2 years in a row. So this is no longer kind of the new, novel thing. It's become pretty widespread. Now, a lot of you, you heard about the Neanderthal, what percentage are you Neanderthal? And you're like, what's the point of that? And a lot of people in the medical community might say, this is not really useful, it's just ancestry. But, DNA, your health, who you are, came from your family. So the best place to start is probably figuring out who your family tree is. So that's a really important step that a lot of Americans have made. And this is really the backbone by which the rest of humanity will understand their source code. And anybody here had their dog tested? You don't have to raise your hands. I won't tell anybody. But yes, you can actually have your dog tested today for a variety of things. I don't have a dog, so I can't say yes. Okay, that was genomics, I'm going to call consumer genomics 1.0. It socialized it. It made it available to everybody. DNA and genetics and genomics is no longer a word that most households know nothing about. They've heard of it. Where are we now moving to? Genomics 2.0, an example of that would be the color test. Color is a startup on the peninsula, and you can, for $99, you'll get a spit tube, and they will test the BRCA 1 and 2 genes, okay? You may have heard about Angelina Jolie. She was one of the people who really publicized clinical grade tests, or what they amount to, widely, partially because of her fame, and partially because of what she did. Now, these genes, there's about, we said there's 3 billion positions, right? BRCA 1 and 2 is about 200,000 positions. And most of them, if there's an error, not a big deal. But some of them, if there's an error, your chance of having cancer goes from the general population average over your lifetime, from 0 to, say, 90, to close to 80%. So this is a hugely impactful discovery if you receive that information. These are probably the most studied genes that we have so far. And the outcome of this can be a fairly big decision. In the case of Angelina Jolie, she had a double mastectomy because for her and her pathogenic variant, it drops her back down to this line. So you can imagine now, as we move from, it's one thing to receive in the email that you're 5% Neanderthal and it turns out you aren't as much, Irish as you thought you were, to get in your Gmail box a PDF that says your risk of cancer might be, 70 to 80%. That's a big deal. Another disease, just to move it away from just the cancer domain, because a lot of people focus on genomics and cancer, rightly so, because cancer is a genetic disease, if you will. Familial hypercholesterolemia. Anybody here had a cholesterol test or a physical? Right? You heard of cholesterol? Heard of that stuff, right? And you've been told, eat more vegetables, eat less red meat, exercise. Those recommendations, let me be clear, are absolutely right. Your health is probably going to be mostly impacted by your lifestyle and your what you're eating and everything else over anything else. But if you have a mutation in one of these 4 genes that we know about so far, your body basically can't process cholesterol, or it's significantly unable to process cholesterol. And no amount of diet or lifestyle change is going to fix that. When you hear about somebody dying of a heart attack at a surprisingly young age, sometimes this is what's going on. What's the treatment? Statins. Statins, I've heard in some cases, again, I'm not a doctor, but if you have a child, let's say, that has this mutation, starting statins pre-adolescent might be the right thing, because if you don't, what happens is you look at the arteries of somebody who has familial hypercholesterolemia, and they're 25, and they look like the arteries of a 70-year-old smoker who's eaten red meat every day. So this also can have a huge impact, and 1 in 200 to 1 in 500 people have this condition. So this is not super, super rare. It's among the most common inherited metabolic disorders that we know of so far. Okay. Now, recently, 23andMe, the FDA finally let 23andMe tell you about these 3 mutations. And there was a lot of controversy about it. People were really happy about it, and people were really worried about it. Let me explain that, kind of disambiguate it. 23andMe, back in the day, because of the costs, they're not testing every single position. What they did is they curated, I believe, about a couple million, 25,000 actually, specific locations that were known to have known something about them, okay? So if you get a 23andMe test, these are the only 3 positions out of 200,000 in BRCA1 and 2 that they're testing. So if the test comes back negative, it doesn't mean you don't have a pathogenic variant in BRCA1 and 2. So imagine, you're a 22-year-old female, your grandmother died of breast cancer, and your aunt has been diagnosed with ovarian cancer, okay? That's a serious family history, and you think, boy, I ought to go get tested for BRCA1 and 2, and you go get a 23andMe test, and it comes back negative. The genomic counseling community said, well, this is a huge problem. I mean, because if you come back with a negative test, that's not a negative test. We've only looked under the streetlight, right? Why am I looking under the streetlight when my keys were lost in the park? Because that's where the light is, okay? Now, these mutations, you might question, like, why are these there? These are called the founder mutations. The Ashkenazi Jewish population went through what's called a population bottleneck, a very small population at one point in time, and a very high percentage of that bottleneck had these mutations. So as a result, these mutations are very prevalent in the Ashkenazi Jewish population, and so they're more prevalent. If you had to pick the top 3, you might pick these. But we have actually a database at UC Santa Cruz that we've built and made available as part of a research project. It's called BRCA Exchange. Anybody can go there, BRCAexchange.org. It's got all the information. It's mostly for genetic counselors and scientists, and medical professionals. But now everybody is starting to come up the curve beyond ancestry into clinical, and we've cataloged 24,584 different variants in just BRCA1 and 2. So that's a huge step up from 3. Now again, most of them are not impactful, but if you do have certain ones of them, it can have a big impact on your health. Now, if you spit in a tube and send your saliva up to color, and you'll get back either one of these 2 PDFs. Hopefully you get the one on the right, and 99% of the time you're going to get that. That says, hey, we didn't find any mutations. Now, the difference from 23andMe was they tested every single position in both those genes. So if you get back a negative test, that means they didn't find any pathogenic variants in BRCA1 or 2, anywhere. If they did find something, then you get this PDF, and it says exactly where the change is. So here a T was replaced with a G, and it's pathogenic. And the next step is probably to talk to a genetic counselor. Now, the other thing about that test, as opposed to 23andMe, is in the fine print. So here's the test, and here's the bottom of it, and it's signed by Annette Meredith, medical geneticist. So it's not, there's actually clinical professionals, and they call this sometimes robo-doctors. There's a big kind of debate in the industry right now with policy and government and medical professionals as to, should you be able to do this without a medical professional? Should you be able to order this test? Now, the way they've done it is they essentially have doctors, and when you sign up on the website and type stuff in, a doctor signs off on the test. The other thing is down here at the bottom, right here. And this says, Laboratory Director Scott Toper, CLIA, and CAP. Those are like ISO 9000 for the FDA and HHS. If a test is performed in a CLIA or CAP certified laboratory, the results a doctor can act on. A doctor could prescribe you statins because of that test. If it's not done in that kind of accreditation, then they can't act on it. They'll order another test. I'm not saying that tests that aren't CLIA or CAP aren't important, but this is kind of where we're moving to. 23andMe wasn't CLIA CAP. These are CLIA CAP. These are the same quality tests. For those engineers in the field, what that means, like ISO 9000, is the clinical process, the chemicals, the way it's done are all highly standardized and reproducible. But also all the software is tracked and all the data is tracked very carefully so that somebody can go back and determine, was it done the right way? Because the outcome of these tests can have big impacts on human health. So let's kind of step back and see kind of where we are. What's your accessibility to these kind of clinical tests? Up at the top, what I've plotted is the number of positions. This is just one way to look at things. Not every position is created equally, so this is not really a great way to quantitatively look at it, but qualitatively it'll kind of help you. 23andMe way back and when, small number of positions. Ancestry is about double. Color gives you BRCA 1 and 2. Color 25, that's 25 genes. A few more. ACMG 59, this stands for American Society of Clinical Medical Geneticists. So it's an accreditation, board certified, and they have curated 59 out of those 25,000 genes. Now, why did they pick those 59? They picked them because we know something about them. A lot of genes we know nothing about. And if we find something about them, you can do something about it. It's actionable. So these are the ones that the medical community most likely is saying, if we're going to test everybody, let's do these. But you can also go up to Invitae, another company in the city that does mostly clinical tests, and you can get 147 genes tested completely. So let's kind of look under the covers for those. Here's the color genes, and I've highlighted BRCA 1 and 2. But if you notice, none of the hypercholesterolemia genes are in that list. Now, if I move over to the ACMG, 2 of those, plus 3 of the 4 hypercholesterolemia genes. And then if I step up to the Invitae 147, I've got all 4 hypercholesterolemia, I'm totally covered, and the BRCA 1 and 2, and then a whole bunch of other genes that are associated with variety of clinical outcomes. So we're no longer, the big change now in the last couple years is we're moving from, picking some places, only looking under the streetlight and looking, around, outside the streetlight. Almost, okay? So one reason why people aren't saying, well, let's just test everything right now, is because we don't know what it all means, okay? So again, BRCA 1 and 2 are the most studied because they started very early, they have huge impact. And if we find a variant, what that means, to be clear here, is there's this human reference genome, which is one person. We're all compared against one person. The current version is HG38. The first one was uploaded from UC Santa Cruz. And if we see a difference there, that's called a variant. About 5% of the time, though, we don't know what it means, which is again, back to that case I sort of cooked up there, 22-year-old female, grandmother died of breast cancer, and aunt's been diagnosed with ovarian cancer. Imagine getting back this result saying, we found something, we don't know what it means. That's that can stress somebody out quite a bit. And that's why there's also a lot of debate as to whether you should be able to have access to that information directly or only given in the context of counseling. 5%. As I move to more genes, the variant of unknown significance rate jumps up to 50+%. Now, why is that? It's just because we don't have enough data. We haven't been in this field long enough to collect and understand what every one of these positions means. If I could wave my hand kind of thought experiment and say, let's take 7.5 billion people, let's look at all those genes, and let's follow their whole history and decide who lived a healthy life and who didn't, then I could probably get this rate down. But right now, it's pretty high. But it doesn't mean we shouldn't go look at it. So this also means this is very different from traditional medicine. Like, how many people have had a blood test? Pretty much everybody, right? Okay. So you had a blood test 5 years ago. Do they call you up ever and say, hey, by the way, we found a whole bunch of new information from that blood test you had 5 years ago? That doesn't happen, right? It's kind of a one-shot deal. But this is data. This is how medicine is being, like every other field, revolutionized with data and software. So let's say we sequence those genes 2 years ago in you. So we know what all the positions are, but we don't know what they mean. And then 2 years later, we go back and look at the latest literature, the latest information, and we go through and we see, hey, have we learned anything new? 10 to 15% of the time right now, when that's done, population scale, not just cancer patients, a negative test turns into a positive test. Now, it doesn't necessarily mean, a positive test you're going to have cancer or other things, but it just basically means there's new information we've learned. So this is like getting that blood test 5 years ago and then getting a push notification saying, hey, by the way, we've learned something new. And this rate, 10 to 15%, this is indicative of how fast the data is flowing and how much we don't know. It's going to go much, much higher. So much so, actually, one of the things we built in the BRCA Exchange is there's a mobile app you can now subscribe to. So again, back to that woman. She gets a variant of unknown significance. Let's say it's this one. She can follow it. And if anything changes, and we update the database, and we do that monthly, she'll get a push notification saying, hey, there was a new research paper published, or a new scientific study, or there was a new case tracked. And all of that data we curate and flow up. That's a pretty darn important push notification, and that's just in 2 genes. Now, how is this happening on a bigger scale? Everybody here heard about healthcare lately? Some debate about it. It's kind of got some problems. Anybody hear about the learning health system? Well, let me ask, how many people know the word closed-looped corrective action? Engineering, manufacturing? No? Okay. Well, recently, Foundation Medicine, they're the #1 cancer testing company in the United States. So if you have cancer and they biopsy your tumor and they send it out, likely they're going to be the lab that tests it. They sequence the tumor, they do a bunch of analysis, and they return back to the doctor a PDF that has drugs. Give this person this, their tumor might respond to this drug. There's another company called Flatiron that actually makes a EHR, an electronic medical record. And last year, Roche purchased both of those companies. Now, why is the interest in all that? Because what they're doing is they're not measuring clinical trials. They're not measuring kind of at the front end that's highly controlled. They're measuring at the back end, what's happening in the real clinical care system. Not at UCSF and Stanford, but at community hospitals. If you went to El Camino Hospital, you got diagnosed with cancer and you were tested, they're tracking that. This is a huge change in the clinical health system. Clinical trials are important. We're not saying that's not the standard. But clinical trials are more to say, does this work? Is it safe? Great, go. There really has not been a way to say, after so many years, what's really working? Or who is it really working for? Because the data does not flow back. It's equivalent in manufacturing. If you shipped 100,000 new phones, you tested the first 1,000 off the factory floor, and you said, hey, the first 1,000 worked good. Oh, we're done. I'm going to go to dinner. See you later. And you just let them roll, right? That's effectively the way the clinical care system works today. Of course you don't do that. You sample ones that are coming back. You look at the ones that are returned. You try to identify defects, and you try to go back and fix them at the next batch coming out of the factory. So this is the beginning of that. Now, we feel strongly this is a great thing, no disregard for what they're doing, but these data, these information is yours, right? All of those 30,000 records that they have are people. And that data is theirs, they generated, but it's also yours. It's society's, it's humanity's. So we're working on programs, for example, with, No, hope I didn't hit the wrong button here. Okay, there we go, we're back. Never mind. So for example, we are consenting, we're doing a pilot study at UCSF where our gastroenterology cancer patients can be consented, and we then go get their raw data and they can upload it and share it, de-identified, to the Ethereum blockchain, so anybody in the world can get at it. Trying to provide provide some pipes to allow that data to flow back. Again, we're all in this together. The outcome of somebody halfway across the world who happens to share some piece of your DNA pattern, and how they respond to a drug or develop cancer or don't cancer will impact your life, if we can get that data to flow. Now, recently there's been a lot of activity at the federal government and the laws about this. Way back when, the federal government put a lot of money into the healthcare system to move everybody to electronic health records. And the thinking was, if we can get everybody electronic, then we can get this data to flow. Didn't work out very well, because what happened was everybody built different systems, and they're all not interoperable. And they also make it really, really hard for you to get your data. Has anybody here had to help somebody or yourself go from one healthcare system to another and move your medical records between them, or even get access to them? It's a nightmare, you know? What if you want to move your photos from your Mac to your PC? It's kind of, you know, you can do it, right? You go to a couple Google and you got it done, right? Maybe take a couple hours. Very hard. So HHS has started saying, no, information blocking means you call up and you say, can I get my data? If they don't make it easy for you, that's called information blocking. And in February, they actually came up with a new set of rules on information blocking, saying, no, you have to actually make it easy to get at it, and if you don't, there'll be stiff penalties on a per-case basis. And back to genetics, what does that mean? What does getting my clinical record mean? It specifically means that they must provide an individual a copy of their complete test report and the full gene variant information generated by the test. You get back that color test, let's say, and it's green. It says we haven't found any pathogenic variants. What that means is we didn't find any variants that we know are pathogenic. But what if they found a bunch of other information that we don't know about, right? If you aren't given back the information that they don't understand, then we can't build a system like that push notification that says, hey, 3 years later, by the way, we learned this new thing, and you might want to make some changes in your life based on this new information. Well, that doesn't happen unless the information goes around. So we're really, feel strongly that this needs to happen. And finally, one of the things we're doing is actually directly with patients. There's groups of cancer survivors. They're typically called previvors if they took a prophylactic step, like Angelina Jolie, to prevent cancer, or postvivors who had cancer. There's a group of about 30,000, and they want to have control over their data. They want to have agency over their data. So we're working with them to develop what's called a data trust. Imagine 30,000 people getting together, putting their data in a lockbox, but then providing access from us researchers and the clinical profession and commercial companies, Roche and UC Santa Cruz, to be able to go in and mine that data to try to get rid of those variants of unknown significance. I don't want to know what any individual person's data in there is, and in fact, one person's data is fairly useless. But 30,000 people's data, that's useful, right? The stats on one of those phones failing could be an anomaly. 30,000 phones, that helps me figure out what's going on. So this is one of the efforts that we're undertaking to allow that, and the key to that is people have to be able to get their information. So as we kind of move into this, next phase from streetlight to sun, illuminating everything, to give you some sense, 1 in 15 will have a high-risk variant. People, 1 in 6 a moderate risk, we believe. Even if your test comes back and says, there's nothing, keep going, you have recessive traits, right? A recessive trait is where one of your genes, has an issue, but because the other one's good, it masks it. Well, future generations are going to receive those recessive genes. So even if you don't have anything, the impact on your kids or somebody else in your family's recessive situation might inform your kids is going to have to be big. For example, when I say population, what does that mean? If we're testing cancer patients, that's a very specific population. But what if I just picked 10,000 random people and I tested them on 139 genes? About 16% of them would have an actionable result. So that's a tiny amount. We're really only just now getting to the point where we're saying, let's test everybody, not just people who have a certain characteristic or family history. If we only test 59, it's about 5.6. Geisinger, should be one Geisinger there. They're kind of the Kaiser of the East Coast, if you think of it that way. They've actually moved way beyond this and are basically just starting to test everybody. The standard of care in England has now become test the entire genome. And they've tested 90,000 people. 60,000 said, yes, tell me something, and they've returned actual results to the doctor and the patient for 1,000 of those. So imagine this, like there's this group over here that's testing stuff, and they email your doctor saying, hey, we found something in a patient that you are seeing. The patient hasn't called you, the patient hasn't seen anything, and the doctor then calls you up and say, hey, by the way, we think you're gonna have a problem. Come on in, let's talk about it. That's completely different from the way we think of healthcare today. So what we really wanna do is test 3 billion on everybody. Actually, it's 6 billion because we have 2 copies of everything. That's really what we want to be able to do, is have everybody have their personal 6 billion, not compared to the reference, not some of it, but the whole thing. And instead of having one haploid genome reference assembly for all humanity that we're compared to, we should have one that's just ours. Now, why didn't we do that to begin with? A little technical digression here. How does this all work? How does that color test work? You would think, most people think, I thought when I got into this, is like, oh, well, they take the DNA and they read it, right? They just put it through something and they look at all the letters on there somehow and they read it out and I get a little text file, right? Turns out that's not the way it worked. They take the DNA and they break it up into millions and millions of tiny pieces, 100 to 300 base pairs long. So imagine I take an encyclopedia and I break it up into strings of words randomly, not even sentences, of 1 to 300, okay? And I just dumped them on you. And then I said, put them back together. That's what the first one was. We had nothing to compare to. And then what we did, when I say the first assembly, we produced a draft. But you see all those gaps? There's whole parts of the genome where we're just dark. We have no idea what they mean. We can't even read them. I'll tell you why in a second. So today, Foundation Medicine, you're a cancer patient at UCSF. We sequence your tumor. We get all these reads out, and then we align it to this draft, right? So we're not really trying to figure out yours. We're trying to kind of hang yours on this other one. That's the way it works today. Amazing. Totally amazing that can be done for less than $4 billion, but it's still not where we need to be. So again, this is like the scientist saying, like, here's all the pieces. I think I found a corner piece. Okay. So, let me see if there's any burning questions before I go on to the 3.0, because we're going to go more technical here in a second.
What is haploid?
Haploid? So diploid, we're diploid. We have 2 copies. And so haploid is, well, there's haploid and haplotypes. So haploid is sort of, there's many definitions. It's ill-defined, actually. But if you're diploid, it means you have 2 copies of the BRCA1 gene. And each of those will have different sequences. And the collection of all the variation in one of those is a haplotype, okay? And really what you want to do, it sets me up for a good thing later on, thank you. What you really want to do is not view like what's the variation in 3 billion. You want to say, what's the collection in the BRCA1 gene that came from mom and the collection from dad? Because when the actual protein's generated, it's a whole haplotype that generates it. Okay. So this is what a high-end Illumina, this is the big, if you're running thousands and thousands a day, this is the high production Illumina, okay? It's an amazing machine. It is mind-boggling that we can go from $4 billion to sequence one person to, you can send it off to a variety of companies, they'll put it in one of these, and for about $1,000, you'll get back your 3 billion aligned to the reference genome. It is amazing. They've done a phenomenal job. But we've seen this story before. Anybody know what this is? Actually, no, I know it's a big computer, but anybody know the model? You win. IBM 360. An amazing machine at the time. Revolutionized business. Made IBM what it is. Versions of it are still running most of the banking system, okay? But these guys came along, right, and said, hey, everybody should have access to this. Doesn't mean the IBM 360 and the Illumina are not incredibly important, but we really want everybody to have access. So 2 professors at Santa Cruz, Mark Atkinson and Dave Deemer, invented a new way to sequence DNA for everybody else. It's called nanopore sequencing. And there's a version of it licensed to a company in Cambridge and Oxford called the Oxford Nanopore, and it's the size of, it's kind of a size of a USB thumb drive or, a disk drive. Literally, that's it. You can buy it for $1,000. It's amazing. That's how everybody's going to get access. So, but it's also not just cost and size, although that's kind of amazing. I don't have one here, but usually Mark, when he gives the talk, he like pulls it out here like this. So imagine I did that, right? It also works completely different than shotgun sequencing in the Illumina. It works the way I as an engineer, or most people think you would want to do it. Here's your DNA. It gets unzipped in a million tiny little things in there. This is called a protein nanopore. It turns out if you want to make super micro stuff, you know, Samsung makes all kinds of super micro stuff. Mother Nature, way better than we've ever been. If you want to make something super accurate and tiny, use DNA to make a protein. So what they do is they make a protein nanopore, and it's small enough to just fit one strand of DNA through it. So literally, your DNA floats around, it unzips, and goes down through this pore. And then what we do is we measure the current across it, and we get out a voltage. We get out a 3 kilohertz, 12-bit file. It's about 1 terabyte to 2 terabytes per human genome at clinical grade. And so we're actually measuring the actual DNA, not some other proxy of it. It's not broken up, and we can get 50K to 1 million long reads out of this, not those 200 to 300. It's portable, so it can be put in a lot of different places. They've put it up on the space station because there's stuff growing on the wall and nobody wants to just haul it back down and figure out what it is. Let's keep it up there and figure out what it is before we bring it back down. We're involved in sequencing the Ebola virus. And they sequence it in the field because nobody wants to get Ebola in the mail, right? You call up. When it first was discovered, it was like, where do we send it? It's like, ooh, oh, no, not me, no. So now they sequence it and they send the data file to a variety of people, including Jim Kent, who was the guy who actually assembled the first human genome. He's the one who's now then sequencing up and assembling Ebola. So second generation versus third. What's the difference other than cost and this cool, shiny technology? Well, I think of it as the first generation is like a 8-bit grayscale image, maybe 640 by 480. It's still amazing that you can take it and have 100 copies of it, but it's still a grayscale image. And what we really want is a 24-bit RGB image at 4K. What does that mean in genomics? Okay. When we sequence those little tiny pieces, what it's really, really good at, what Illumina is very accurate at, is finding where one position changed to another. It has a very low error rate. And back to those BRCA variations, there are pathogenic variants where one letter got changed to another letter, and that has a big impact. But there's also these bigger things called structural variation where genes move around and nucleotides change and they get inverted and large structural rearrangements. These happens all the time. If you're writing software, right, how often do you just change one letter and how often do you move like a variable in a line? You think about, if I said the only thing I can track is changing one letter in your source code, we'd be kind of limited. So because we get the long reads, we can actually measure these big variations. So all of a sudden, there's a huge piece of human variation that all of a sudden we can actually measure. The other thing is we can actually, because of those things, we can actually say, back to your haplotype question, we can say this all came from mom and this all came from dad. That's called phasing. And it turns out, more or less it's 50-50, but sometimes you can get more from mom, and sometimes you can get, more from dad and less from mom. And we know anybody whose parents are like a big family, you say like, these two kids look almost identical. It's because they probably got more genes, more DNA from one parent than another parent. So for the first time, then we can see them all together. It's interesting for those reasons, but it's more interesting so that you can say, oh, this parent had this clinical history, this family history. And here's the child. Well, what from that parent did the child get? As opposed to right now, it's just a wild guess. And you have to go test the parents and the rest of the family to figure this out. It's called a trio. Now the other thing is when we said that the human genome has all these genes in it and there's all these others that are regulation, well, Mother Nature has another way to control all this. One of the ways is called methylation. So when you're born, the gametes, the methylation stripped off. And then as you go through your life, pieces of your genome will become methylated. What does that mean? Here's your DNA, and these little methyl groups get attached. And what they essentially do is they prevent the DNA from being transcribed or expressed at those positions. It's like on and off switch. Think of you've got a million lines of source code. Is it all running? No. Some of it is running depending on what you're doing. Some of it's turned off. And this has huge impacts. It also has a bunch of interesting societal issues. There's been some studies that they correlate socioeconomic status with methylation. If you may have heard someone, which are still under debate, if somebody in your family went through a famine 3 or 4 generations back, that can be passed down through epigenetics is the term. 14,000 scientific publications talk about methylation. There's only 14 commercially available clinical tests. So this is like, we've got all these pictures, but the whole red, like they're all blue and green. The red we don't see. We can't even see it at all. Well, nanopore sequencing directly measures this because we're actually directly measuring as the DNA goes through. If it's methylated, the signal will have that information in it. Now, as I'm a machine learning person, I find this incredibly fascinating because let me kind of spell this out. Remember we talked about this learning healthcare system where you got this push notification that says, hey, we did this test 3 years ago, and now we just redid the data file and we found there's some information and you might want to do something. So let's say we sequence your DNA using a nanopore and we store the original terabyte file, the signal file. And then we discover some new modification other than methylation. Well, the way we figure out what this signal is to DNA, to ATCG, is we use a recurrent neural network, a deep neural network, right? Everybody's using a neural network for everything today, right? Well, we train it. And as we start to get more data, we improve the neural network. So imagine I'm a healthcare system, and I have 100,000 people under management, and I have their 1 terabyte file sitting on a disk that I measured, let's say, for the sake of a discussion, 5 years ago. And now, the latest version of the neural network that's measuring methylation and 2 or 3 other things that we didn't know about. I then run the neural network in the forward inference direction on all those terabyte files, and I will come up with new measurements. I will be able to measure your biology in the past now, which is amazing. It's basically Back to the Future. Wrapping doctors' heads around this is really hard. Wrapping machine learning people around it is like, yeah, of course, why not? So this is really groundbreaking. It's really going to change the game. So we, last month actually, to kind of really finally do this for real, we've started a pilot project that is going to expand quite significantly in the near future. We sequenced 11 reference genomes in 9 days. Now you might say, like, is that fast or not? That's blindingly fast. And reference quality genomes are significantly higher accuracy than clinical grade. We did them all in this. So that little tiny one I showed you is this one right here. This is called the Prometheon. This is their server workstation, right? It's still the size of a laser printer, but it's what can go up against an Illumina. And you can see there's basically like 48 of these in there. And what we're able to do is measure. So we didn't choose anybody. We chose, there's a project called 1,000 Genomes where people from various populations were sampled, sequenced, and we chose I think it was 10 or 11, to represent 97% of humanity as we know it. And then we pulled out of it their individualized assembly. We didn't compare them to that old reference genome. We sequenced them personally, individually. Their structural variants, phased, mom and dad, and their methylome. Now, where is this going? Instead of a reference genome, right, comparing everybody to this one fictitious person, we're building what's called the graph genome. So picture all of humanity. Any one of you is on one of these streams through this graph. And if you're on the red stream, there's hundreds of thousands of other people in that red stream. The fact that you differ from the green stream is called, humanity. That's diversity. That's life. But then what we can do is we sequence, we add you to this, and we say, oh, everywhere you're the same except for right here. You diverge from the blue stream significantly right here. That's where we should look. That's what can cause disease, or might give you superhuman strength. Another topic we can discuss later on. So this is really, the future of genomics 3.0 is really the personalized, where we can measure you deeply, all of those aspects, come up with your reference to you, and ultimately unlock life's code for everybody. Thank you.